Welcome back, guys. So this is the first session of our neuroanatomy. I will start with the spinal cord. So I know you guys are quite not happy or you don't like a neuro subject, but trust me, if you understand the basics, this is the most interesting subject you will ever find in your medical sciences. So this is my favorite area. I love reading about it and I love teaching about it. I hope I may be able to transmit the same infection of loving the neuro subject to you guys. So with that hope, I'll start the subject. My only one request to you guys is leave the fear outside. Just be neutral. I'm not asking you to love the subject. Just be neutral with an open mind. Listen, try to understand, and again try to recall again after the sub portion or topic is over. This beautiful topic. Okay. So spinal cord. It is. The starting part will start with this, but before that I will give you a couple of slides with some introduction, and then I'll take you to it. You might be aware of most of this part of it. So I'll be doing spinal cord into two parts. This part will do till the gray matter and the tracts and the blood supply and the applied aspect we'll be doing in the part 2. So let us begin. So nervous system. So we all know nervous system is, so here it is self-explanatory. Wherever any explanation is needed, I'll get into it because I have spent quite a considerable amount of time in making this presentation. So it is very much self-explanatory. If it was a class, since you will be in front of me, I have a liberty to talk or give example and draw so many things. But since it is a pre-recorded lecture, so I have done everything from my side to make this very interesting, and I have put every slide into it. That's why the number of slides have gone up, but it will definitely make your job much easier. Okay then. So nervous system. So here we know a nervous system is made up of cells. The electrically excitable cells is what we call them as a neuron. So these are your neurons, which are your electrically excitable cells. And then similarly, the electrically non-excitable cells are your neuroglia. And if you remember from my introduction to nervous system, so I mentioned there that your neuron contribute around 20%. They contribute around your 20% of your cell population in the entire nervous system, whereas the neuroglia will contribute the remaining 80% of it. So thereby your neuroglial cells are more in number compared to your neurons, your supporting cells. Your neuroglial cells are your astrocytes, oligodendrocytes, microglia, ependymal cells. Those are the example of neuroglial cells. And we also dissolve everything, all this material that is neuron and neuroglial cells with the blood vessels and the myelinated and non-myelinated nerve fibers and the supportive connective tissue, all are grouped together, put together, and they are covered by your coverings, meninges. So they will be covering and protecting them. So entire brain and spinal cord are covered by them. So the one which are covering your brain, you call them as a cranial meninges, like a cranial dura, cranial arachnoid matter, and cranial pia matter. Same way, the one which are covering your spinal cord, you call them as a spinal dura, spinal arachnoid, and spinal pia. But then they are continuation from the cranial to the spinal. We'll be knowing in this detail later on. And we know that neurons and neuroglia put together, and if they are present inside the CNS, we call them as a nuclei, or it is also called as a gray matter. Same thing, if the neurons or neuroglia they are present outside the CNS, we use the word as a glial cell or your ganglia, the ganglia. So you have the ganglia in your spinal nerve, that is what we call dorsal root ganglia. Also you may heard the name as a your dorsal root ganglia, or also called as spinal ganglia. The other ganglia, if you are aware, in your GIT system you have those plexuses are there, myenteric plexus, Meissner's plexus. Those plexus we have this parasympathetic and sympathetic ganglia, like superior celiac ganglia, superior mesenteric ganglia, inferior mesenteric ganglia, celiac ganglia. So many ganglias are there. So these are your sympathetic and parasympathetic ganglia. So those are the ganglia which are outside again your nervous system. They are nothing but the collection of your cell bodies outside the nervous system. In the same way, the fibers of your neurons, and your fibers of your glial cells, they form your nerve fibers. Now within the CNS you use the word as a tract. Same thing outside, if you see, you call them as a nerve. So nerve, your femoral nerve, tibial nerve, your ulnar nerve, radial nerve, median nerve, axillary nerve, all these things, if you take them back into the spinal cord in the reverse order, they will be a part and parcel of some or the other tract. They will be part and they will be receiving fiber from both ascending and descending tract. So that's about your nervous system in brief. So let us classify the nervous system. So nervous system can be classified into, this is only for our convenience, into a part which is lying in the center called central nervous system, and the part which is lying not in the center but at the corner, we call that as a peripheral nervous system. The central nervous system is your brain and spinal cord, whereas the peripheral nervous system is made up of your somatic nervous system. So I can write this system as a somatic nervous system, which in turn is made up of your cranial nerves, which are 12 pairs. And cranial nerves are the nerves which either come out or goes in to the skull and into the brain. And spinal nerves are the one which are 31 pair. They come out or go in via your foramina of your vertebral column. So it is something like they are the wire from the spinal cord and they are the wire from the brain. So as far as the spinal nerves are concerned, all spinal nerves are mixed now, whereas a cranial nerve, they are got sensory, motor and mixed. Now why we call the system as the somatic nervous system? Because the somatic word comes from the word soma, which means body wall. So here it is via cranial nerve it is supplying your head and neck, and via your spinal nerve it is supplying the rest of the body wall. So that is the reason the entire both these nerves together are supplying your entire body wall or the structure present on the body wall. That is why it can also be called as a somatic nervous system or craniospinal nervous system. And on the other side we got the autonomic nervous system. So autonomic nervous system is an automatic system which is not in our control, and it supplies the smooth muscle and the cardiac muscle present in the body. So cardiac muscle, we know it is a myocardium present in the heart, whereas the smooth muscle we have got elsewhere. Like your example of smooth muscle will be your smooth muscle which are present in your artery and veins, so blood vessels, smooth muscle present in your glands, that is sweat gland, sebaceous gland, all the glands, all exocrine glands, so that smooth muscle, and your erector pylori, that is your involuntary muscle what we have in the skin which gives us the goosebumps. So all these smooth muscles are again controlled by your sympathetic and parasympathetic nervous system. So this is in brief about your nervous system. So the neuroanatomy class will be concentrating on this part of your nervous system. That means we are doing the central nervous system. So exactly your neuroanatomy is nothing but central nervous system. Now whereas the peripheral nervous system, what about the peripheral nervous system? The peripheral nervous system we have already done region wise. If you remember, so we have done the cranial nerves when we are doing the head and neck. So head and neck we have done the cranial nerve, and then as far as the spinal nerves are concerned, this we have done region wise. So upper limb we have done the upper limb spinal nerves, then lower limb spinal nerve, then thorax intercostal nerves, and so many nerves we have done in this way. So that is how we have even done the sacral nerve and coccygeal nerve in your pelvis and perineum. So those nerves, peripheral nerve, we have already finished in this way. And autonomic nerves are the one we have done when we are doing our thorax and the abdomen viscera and the pelvis perineum viscera. So when we did the viscera, we always mentioned that the supply being a sympathetic and parasympathetic. So thereby we have done that part of it also. So the part which we have not touched is your brain and spinal cord, and that is the reason this entire portion we will do the neuroanatomy. From the university point of view, your neuroanatomy weighs around your 12 marks, but this is not hard and fast rule, as you all know. So last June, July, the main exam, they have asked more than 15 marks, and all the questions were very tough. That is the reason you all people are aware of what happened in the last. We usually call that as a massacre. So massacre. So it's a neuroanatomy massacre which happened in the last batch. So many of you have been the victim of this massacre, So we don't want this massacre to happen ever in your life again. So we'll take care of that part of it. You will love the subject. Many students who have attended the sessions, they definitely or they started liking it, and they have even told that they want to become neurosurgeon or a neurologist or a psychiatrist because the subject itself is so interesting. Okay, so that's all about the introduction. So let us talk again in the brain and the spinal cord. Here we will again ensure that we are talking about in this first we will start our learning with your spinal cord, and once the spinal cord is done, then we'll proceed up, because spinal cord is the simplest form of your central nervous system. So once we understand the simplicity of the spinal cord, then we can build up the difficult concept up there. So later we'll be doing the part of the brain. Again in the brain also we'll be starting from the lower to up, which include your, as the spinal cord comes up. So if I include one more page here, so it is something like the plan will be, so we'll be doing, first we'll do the spinal cord, and from spinal cord then we are going up, and that will lead us to brainstem. So brainstem is again made up of three parts. Then from below upward it is your medulla oblongata, then you got pons, and then midbrain. So that means once we reach the midbrain, we say the brainstem is over. Then we enter from the brain. Now brainstem is done. We will enter now in the next topic. In the next topic is your diencephalon. So that is your diencephalon. The word looks quite dangerous. It looks very, what is it? It's like a bomb, RDX. But then it is a word, it literally simply means it is a collection of nuclei, gray matter in and around your thalamus. So that here we will learn about thalamus. So this is all, the whole family is called thalamus, epithalamus, hypothalamus, metathalamus. So thalamus, epithalamus, hypothalamus, metathalamus, and all this, and subthalamus. All these thalami together is what we call as a diencephalon. So when we are doing diencephalon, once the diencephalon is done, we move up there. So now beyond this we'll be doing our what we call as your, after this diencephalon we'll be doing our telencephalon. So telencephalon is the highest part of your brain. So here we will talk about our cerebrum. So here we'll talk about our cerebrum in detail about this. So this is how the whole plan will be. Now there are other words are there which we use. So I will talk about what is this telencephalon and diencephalon. These words I want to give an introduction to it. I will talk about it. And before that, we will also talk about, when we are doing this, we'll also mention that when we are doing the brainstem, we'll talk about our fourth ventricle. So ventricular system also is present. So your brain is nothing but, brain and spinal cord is nothing but your gray matter, white matter, and no matter. No matter means the empty space filled with the CSF fluid. So fourth ventricle will be done when we are doing your brainstem. So along with the brainstem, when we are doing fourth ventricle, we'll definitely add a cerebellum also to it. So that is the concept of your midbrain, hindbrain, and forebrain. So I'll come to that part also. And then when we are doing the diencephalon, so that time we'll look at the third ventricle. So third ventricle is a cavity of your diencephalon. When we are doing telencephalon, we will do our lateral ventricle. So your basal ganglia is also part of your cerebrum, and your basal ganglia, they all are your part of your limbic system, all part of your telencephalon. Even the blood supply also will be done as and when we are going to this part. So if you look at the concept, there are two ways we can, first if you see the term which we use, so we have the word called as a forebrain. Fore means in front, forward, we say forebrain, or fore mirror, hind mirror, forebrain, or foreleg and hind leg. So forebrain. Forebrain include anything which is in front, which is lying in the anterior part or front part. So this will be mainly your cerebrum. cerebrum plus basal ganglia plus your limbic system, whatever. And then if you move down, then we have got, and even this also forebrain also include your diencephalon, which means all your thalamus, hypothalamus, whatever I told you, all of it. And then there is a midbrain. So midbrain is the part which connect your forebrain to hindbrain. So the name itself says it will be in the middle. So this portion here will be talking about your substantia nigra, red nucleus, then your cerebral peduncles, and your tectal nuclei and all, so on and so forth. We'll see this later. So this is what, at least you know the substantia nigra here in this part. Then we go down, and then we have got our hindbrain. Now hindbrain means behind. This is before, that is how the word before come. This is behind, that is hind come. That means it is a posterior most part of the brain. And here we'll talk about your brainstem and cerebellum. Brainstem and cerebellum together, except midbrain. So brainstem, here we will not talk about the midbrain because it has already been covered, except the midbrain. So that means pons and medulla will make a brainstem along with the cerebellum. So thereby, in the same way, if you want to say the ventricular cavity, the hindbrain ventricular cavity is your fourth ventricle, midbrain ventricular cavity is your third ventricle, and your forebrain ventricular cavity is your lateral ventricle. Yeah, so now what about this, the concept of diencephalon and all? So before we go to this concept of diencephalon, I have to tell you something little bit about the development. So the development, if you know, so development, this is an overview of it. So development of your central nervous system. It is just, I am avoiding all the detail, it is already been done in embryology. So just giving the overview of this. So to begin with, all your systems in your body are just like tubes. Just imagine like there are straws, the thick milkshake straws. There are around 10 to 12 straws are there within your body, and they are made up of your germ layer. And one of the straw is what we call as a future nervous system. So I will call that straw as like here. To begin with, it is something like this. So this is a straw which is there, and you can call this as a your neural tube. So that's your neural tube which you have got. Now this neural tube, I'm not looking at the timeline and all. So neural tube has got an upper opening, this we call as a cranial pore, and the lower opening called as a caudal pore. So but before the neural tube, how it was. So if you may remember that before this you have got a plate. So there is a plate is there, procaudal plate, if you remember, the tennis racket kind of thing. So here there will be folding will happen on this side and this side. So ultimately when they fold, it is something like you got a paper of this shape, and you fold both the end, and then it will lead to formation of a tube. So from a plate you got a tube. Now this tube will further develop now. So this development happened because inside them you have got the germ cells. Inside them there are stem cells are there. Inside the wall of the tube there are stem cells are there. Now these stem cells, they keep on dividing rapidly, and thereby we have the development of the nervous system. So this development doesn't happen at a same rate. So that to begin with, this is just a neural tube to start with, and later on the neural tube, inside the cells will divide rapidly. Some divide very rapidly and some divide slowly. So thereby the entire system now will develop into, so if you see, so in this again, especially this part, if I say your upper 2/3 area of the neural tube, the division happens very rapidly, cell division, whereas your lower 1/3 area, the division happen slowly, but they are dividing. So the growth is happening in both the end. So the lower part is what we call as a caudal part of neural tube, whereas the upper portion, the division is very rapid. So thereby what happens is it develops into, it gives rise to I'm just looking for a picture. Okay. Now, because of the development in the upper part, so now the next thing which develop out of this, and there will be a closure. Yeah. So before we begin there, now what happens is your upper end will close and the lower end also closes. I think around 26th day it closes, and this closes 2 days later, 28th day. And if this failed to close on 26th day, that lead to anencephaly. You may remember the skull may remain open, anencephaly. And if this failed to close, then it may lead to your spina bifida. So this is all your development. Anyway, so this is closed now. So now what happens is, now there is a development, is division inside the tube is happening, and the division is upper part divides very rapidly, then middle part it is okay, then lower part also divides, and this is your caudal part of neural tube as it is. So this one is your lower part which I was telling. So this is your lower 1/3. We can call this as a, so thereby lower 1/3 division is very slow, whereas upper 2/3 the division has happened rapidly. So inside this you got all your cells are there. These are your cells which are dividing. Here also cells are there. They're dividing very rapidly. Now here we have got, now it has given rise to now 3 different extension or cavities are there. So they have been labeled now. The upper extended part is what we call as your prosencephalon. And this middle slow dividing part is what we call as a mesencephalon. And the lower part above your caudal part of neural tube is called as your rhombencephalon. Now prosencephalon, mesencephalon and rhombencephalon. So prosencephalon, you split the word pro. Pro means something which comes before. Procurement, producer. Pro means before. Prosen. Cephalon means brain. So it is cephalon, cephalitis. So this is brain and this is before. So that is, this is the part which is actually forming first. Mesen means something which comes in the middle, in the timeline. Cephalon is again the brain. And same thing, rhomben is behind. It is a behind brain, or the last part of the brain to come. So if we want to give the name, the prosencephalon is also called as forebrain. This is the one which will develop all the derivative of forebrain. Mesencephalon will become your midbrain, and your rhombencephalon will become your hindbrain. So these are the 3 parts. Now remember, the development is still happening, it is not yet over. Now this part, that is your prosencephalon, mesencephalon and rhombencephalon, and even including the lower part. So this portion what we are calling caudal part of neural tube. So everything is neural tube, but this portion is dividing very slowly, so we still call this as a neural tube, caudal part of neural tube. Now this caudal part of neural tube will form your future spinal cord. Okay, now from here, now the development has to proceed now. So what happens now? Now in this area, especially the prosencephalon, you see in this also some part, within the prosencephalon, some part will divide very rapidly, and some part, like this area, will divide not so rapidly. Similarly, in your rhombencephalon also, some part divides very rapidly and some part divides not so rapidly. Mesencephalon having its own rate of division. So this also will give rise, because of this difference in the division and multiplication of cell, now we have a altogether a different structure coming out of it. And before that I go there, so here you can see now, because here we have prosencephalon, mesencephalon and rhombencephalon, and you can see in the center of it, the entire center portion, if you see this part where I am putting the cross mark, or let me use a different color. So this portion. So that is still empty, and it is what is the future ventricular cavity. So there is a ventricular cavity in prosencephalon, there is a ventricular cavity in mesencephalon, there is a ventricular cavity in rhombencephalon. So now this stage, at this particular stage in development, because there are three extensions have formed. So this is what we call as a, and they are first to begin from the neural tube. They are called as a primary vesicle stage. Vesicles, secretory vesicle, some dilated part. So this is your primary vesicle stage where three vesicles have formed. Now the development continues from here because of this, where the development is rapid in some part and the slow in some part. So this will give rise to something like this. Because of lack of space, I'm just putting it to, yeah. So here we have this, and remember our cells are still there as it is. They are not gone anywhere. They are still there as it is. These are your stem cells which are still dividing because development is going on even after birth also, at least for the first 5 or 6 birthdays, or some say even the first 10 years of your life, the brain is still developing or dividing. So these are your cells, and there is a cavity. Whatever the white part is there, that is your ventricular cavity as it is. So that's your ventricular cavity as it is. Okay. So now prosencephalon. So this one was, we have called this portion as your prosencephalon. So this part is your prosencephalon, and this part, the small narrow part, is your mesencephalon as it is, and this portion is your rhombencephalon, and this narrow part is your caudal part of neural tube. So now you see a new structure has come up because of the differences in the development or division. The upper prosencephalon, the rapidly dividing part, is what we call as this rapid dividing part is called as, this portion which is dividing very rapidly is what we call as a telencephalon. The word telen word comes from the word tele. Tele means telephone, telegraph, telepathy, telomere. Tele means something which is far. So it is a far growing or growing very fast, so it is going away from it. That is why it is called as a far brain. Far brain or going farther brain. And a part which is again next to it, but it is dividing very slowly. This portion, this part which is there, and next to it, this portion, is what we call as a diencephalon. Now the diencephalon and prosencephalon, telencephalon and diencephalon together, we can call this as a prosencephalon. That's how the word prosencephalon came. And the mesencephalon is as it is. That's your mesencephalon, which has grown, but no further growth has happened. No differentiation much. The rhombencephalon, you see, the upper part of your rhombencephalon is what we call as a, where the division has happened rapidly, meten, M-E-T-E-N, metencephalon. The same way, there is even a lower part where the division has happened rapidly, leaving the center part where the division is not so rapid. That is why that curve came. This we call as a myelencephalon. So there is a metencephalon and myelencephalon together. You will call this as a rhombencephalon. So thereby we have got now prosencephalon, and then metencephalon and rhombencephalon. So prosencephalon is divided into telen and diencephalon. Rhombencephalon is divided into cranial metencephalon and caudal myelencephalon. And the lower part is as it is. It is again here, I'll call this as a caudal part of neural tube, as it is, caudal part of neural tube. Now with this, now what telencephalon will give rise to what diencephalon. So what are the derivatives of these cephalons? So if you see this, now these are, now we are looking at the derivatives. So these are the derivatives of cephalons, whatever we have seen in the previous. So if you see your telencephalon, will develop into your cerebral hemisphere. Yes, of course, along with this you have got your basal ganglia and all. I'm not putting that in limbic system, but mainly here you have to remember this as a cerebral hemisphere, because within the cerebral hemisphere only we have got these portions. Anyways, and here we have the ventricular cavity, which is your lateral ventricle. So if you go back in this, and if I have to write the name of the cavity in this, so this is here one lateral ventricle on this side and another lateral ventricle on this side. And whereas in your diencephalon you got your third ventricle. And your mesencephalon, this will be a cerebral aqueduct. It is only a duct which is forming. It is a connection, a duct. Whereas your fourth ventricle will be your cavity of your rhombencephalon, and the central canal will be the cavity of your spinal cord. So here what you got, this one is your lateral ventricle. Then you got telencephalon, and after that you have your mesencephalon. Not mesencephalon, there is a diencephalon, is it? Telen, and then you have a diencephalon. Now, diencephalon and telencephalon together are what we call prosencephalon. So this will give rise to all your thalamus, metathalamus, hypothalamus, subthalamus, and epithalamus. So what are each of it? At least thalamus and subthalamus and hypothalamus you know from your physiology point of view. Your metathalamus is nothing but your medial and lateral geniculate body together we call metathalamus. And subthalamus is the connection between your midbrain and your thalamus. We will see this later. And epithalamus include your pineal gland and all. Anyways, details will come in future, but right now these are all the derivative from diencephalon. And what is a cavity here? We said it is your third ventricle. Next, so this both are together if you put in this one box. So this one is your, all this is a derivative of your prosencephalon. In other words, forebrain. So the forebrain cavity is a lateral ventricle, and forebrain is all this. Whenever we use a forebrain, we mean this. Next is your mesencephalon. So mesencephalon is the part which is growing very slowly among all your cephalon. Mesencephalon give rise to midbrain. So it forms your midbrain. So in midbrain we will be talking about your substantia nigra, red nucleus, so many things are there. We'll see this later, but that's just to give you example. Tectum, tegmentum, and all these are present in part and parcel of here this thing. And here we have no proper ventricle, but only the one which is connecting third ventricle to fourth ventricle. We call this as a cerebral aqueduct of Sylvius. Just a tube which is connecting your a ventricle lying above third to the ventricle lying below called fourth ventricle. Now next is your rhombencephalon. Rhombencephalon, we said it is divided into two parts. The upper part is your metencephalon, and lower part is your myelencephalon. So metencephalon will develop into your pons, and very important, please pay attention, cerebellum and not medulla. That means pons and cerebellum are siblings, whereas your myelencephalon will give rise to your medulla oblongata. And the cavity which is developing in your entire rhombencephalon is your fourth ventricle. Then the last part is your caudal part of neural tube. And this will give rise to your spinal cord. There is something called as a LR plate and all, so I will not get into the details of this, so which include again the LR plate, matrix zone, mantle zone, so many things are there, so I will not get into that. So spinal cord. And what is the ventricle of the spinal cord? We call simply as a central canal, because it's located in the center of the tube of spinal cord. So if you come back to this, so in this particular stage, if you count, you have got 1, that is your telencephalon 1, diencephalon 2, mesencephalon 3, metencephalon 4, and myelencephalon 5. So total there are 5 vesicles have formed here. So this particular stage is what we call as secondary vesicle stage. So there are 5 vesicles in the secondary stage. These are your developmental words. Secondary vesicle stage is 5, primary vesicle stage are 3, prosen, meson and rhomben, whereas secondary is your telen, dien, meson, meten, myelin. So these are all your part of it and development of it. So this is in detail, somewhat in detail about your development of the nervous system along with the development of ventricular system, how it is formed. Fine. So that's all about, in brief, about the nervous system and its development and how we get these words. So I'll be using these words simultaneously. So here when we were talking about the part here, that is what I was telling. So we are now doing here spinal cord, which is nothing but the lowermost part of your neural tube, caudal part of neural tube. So, and thereby here we will have our ventricle will be your central canal, where your CSF will be flowing. And whichever any part if you take, if you imagine the every part of your brain or entire brain as your cauliflower with a stem. So that stem will be your brainstem. And if you take the stem little longer, and that will be your extension of the stem, we can call spinal cord. And both diencephalon and telencephalon together as your actual cauliflower. And if you chop from the bottom all the way, it is something like this. So if you take this, I'll just take this image. If you take this image, and if you chop this, and if you're cutting it like how you will cut a cucumber, so then if you're cutting it anywhere in the spinal cord, anywhere, we will see the details of this, or in your medulla, or in the pons, or cerebellum, or your midbrain, or anywhere in your entire cerebrum also. So these are the transverse section when you cut. So when you cut this, you will find, as far as your brain is concerned, the entire brain, that is all the yellow part, here you will find there is a central, you will have centrally located white matter will be present, which is white in color or pale in color, whereas the peripheral portion around it, you will have gray matter. Now there is an exception to the central white and peripheral gray in the brain, exception being brainstem. So brainstem, except brainstem. So brainstem will have the reverse. That means it will have same like your spinal cord, except. So brainstem, except brainstem. When you do the spinal cord, the entire spinal cord plus brainstem. Remember what is a brainstem means? Brainstem, I told you, in medulla oblongata, pons, and midbrain. This is, so if you do take a section here, so here what you will find, it is exactly opposite of this. So here what we will have a central gray matter, butterfly shaped gray matter, whereas a peripheral white matter. So this is, so that means you can say your brainstem and your spinal cord more or less structurally, in terms of arrangement of your gray and white matter, it is same. So whenever the word gray and white comes from now onward, so whenever we use the word gray matter is made up of cell bodies of neurons. Neurons are your electrically excitable cells. Cell bodies of neuron and your unmyelinated. So unmyelinated are the fibers which are not being coated with the sphingolipid or a kind of a fat. Unmyelinated nerve fibers. Nerve fibers are your axons and your dendrites. So this is, which is what we call a gray matter. So apart from the unmyelinated nerve fiber in the cell bodies, you also have your glial cells, neuroglial cells. The reason is there are 80% of cells, so they have to be present here also. And all this need nourishment, so you also will be having blood vessels, both arteries and veins in this. So this entire package, you will call this as your gray matter. Now similarly, if you look at what is a white matter, so white is called as a white matter is, here it is made up of myelinated nerve fiber. Now here you have that lipid which actually will coat all these electrical wires called as a axon. So myelinated nerve fiber, so that will give a appearance that it is brighter, that means slightly it has a whiter shade compared to the darker shade of the other part. And since the glial cells are there 80%, they are also present in your white matter also, and then blood vessels. So to understand this, easy way will be again here cake. If you look at the cake, if cake is your brain, the upper cream part, this is what we call as a cream portion of the cake, and this is the bread part of the cake. So the bread part of the cake is your white matter. So much is there, almost you can say white matter is around 80% of it, whereas the cream part of the cake is your gray matter. And now when I say gray matter, what should come to your mind? All these things. And when I say white matter, all these things. So this is what is your entire brain made up of. And if you take a slice out of it, you take a small piece out of it, you will always find, if you take a small slice from here, so again this also in the top layer will be your gray, rest of it will be your white. So this is what we do the histology. So in histology of cerebrum, what is done, they take a small chunk of slice like this and they do the histology. So in this layer you will find 6 layer of cerebrum, all the 6 layer, and this is your white matter. Yeah, okay, histologically. So this is all in brief about the introduction. So let us bring, now we will talk all about our spinal cord. You understand spinal cord, understanding of your spinal cord is very important to understand the brainstem, because it is something, the most simplest part of the nervous system is developing from spinal cord and then it initiates. So thereby some part of the spinal cord still retain in the brainstem as we proceed from the medulla oblongata, pons and midbrain. So let us talk about your spinal cord. So it is very simple, wherever explanation is needed I'll give you. So now spinal cord can be compared with, it can be compared with the input and output system of our computer. So what we have, input is something like you type something, and if you give a print command and whatever comes out from a printer, that is an output, or whatever the software coming out, or whatever the command gives and whatever the information comes out, so that is what is an output. So your spinal cord receives the information and it sends the information to the brain react to that information and then send things back, response back. So that is why we can call this as a input output system of a computer. Now in terms of the physical appearance, it is elongated, quite a long tube, cylindrical, it is like a snake, cylindrical, though it is flattened anteroposteriorly, part of the CNS. So because your CNS is made up of brain and spinal cord, and the part which is elongated and cylindrical, we call that as a spinal cord, and it is present within the upper 2/3 of your vertebral canal. Vertebral canal is your backbone, and it is something like this snake you have taken, and you have taken the iron box and you have ironed it. So what has happened to this, it has got flattened anteroposteriorly. It is not cylindrical like a tube, but it is flattened anteroposteriorly. It is something like it was there on the floor and by mistake you stamped it with your foot, so it became flattened. Now what are the functions of the spinal cord with this input and output? So it receives various modalities of your sensory input, like your ascending pathway, your touch, pressure, pain, temperature, vibration, stereognosis, all of this are your different sensory modalities. So you are receiving through the ascending pathway because your body is receiving wherever it is touching. From there the information is going to spinal cord, and from spinal cord information is going to your brain via the ascending tract, and the brain is giving the information back to your spinal cord via descending tract, and via descending tract it is coming to the nerves and to the muscle. That's how we react. So thereby different modalities of sensory input. Then this sensory input is associated. This information is associated with your different parts of your brain and spinal cord and produces a reflex response in reply. That's what you receive, you work on it, you compute it, and then you produce a response. So this is what is exactly the spinal cord does. So but if you want to say that it is not always the brain is involved, so spinal cord is also involved in some functions where you don't need the intervention of the brain. So they are also called as a spinal reflexes. So what the spinal cord does, apart from taking the information and giving back to the brain, it also executes simple reflexes. So apart from this, the other functions include execution of simple reflexes, where you don't need involvement of the brain. Other than this, it is as simple as, if you want to keep it as simple, so it means it help in passage of transmission or passage of electrical current, which we call as an impulse. So passage of electrical current to the brain and back from the brain, from brain. So this is what is as simple as we can put it. Now let us talk about the extent. For physiology guys, this is important. You already know the tracks, ascending and descending tracks and all, but this all will make up because it is anatomy. So once you know the structure and form, it is very easy to understand the function of it. It is something like anatomy is like a theater. Physiology is a movie running in it. So if you understand the theater very well, you can enjoy the movie also. So bear with me until we go, at least for the first part of it, it is mostly anatomy. In the second part, it will be mostly physiology. So what is the extent? From where to where it extends, this snake? So it is in a continuation of your medulla oblongata. We know here, you can see in the picture also. So medulla oblongata, the moment it comes down from the foramen magnum. So you can see here, the moment it is coming down, this is a medulla oblongata here in the picture. The moment it comes down, you call this as a spinal cord has begun. So thereby, to assign a value, you can say upper border of your C1 vertebra. So your upper border of your C1 vertebra, which is also called as atlas. So that is what is the beginning of it, or else you can also call as a lower margin of your foramen magnum as a upper extent. It goes all the way down there. So lower it is where it ends, it is variable. So in case of your, you can see here, it is ending in this case at L1 vertebra. Yeah, and this L1 vertebra ending is happening in the low, that is what we call lower border of L1 vertebra. This, or even sometime we can say otherwise, saying the intervertebral disc between L1 and L2. So this is have seen in your adults, whereas in case of your infant, a newborn, it is at L3 vertebra. This is important. So this is the difference is there. So why this difference come, I will talk about it. So thereby, from C1 vertebra, it is common above to your L1, L2 vertebra in adult and L3 vertebra in case of infants. Now when it is ending at lower end, you can notice that it is ending as a cone instead of ending bluntly. It is ending like a cone. It is like this, it is ending. Now this is what we call as a conus medullaris. Now from this tip or apex of the cone, we have a thread like filament which is passing down, and that thread like filament is what we call as a phylum terminale. So this is, so thereby the end of the spinal cord will be the conus medullaris. So conus medullaris is the end of spinal cord. From there, your pia mater, which we call as the phylum terminale, it extends downward. So that is not the extent of your true extent of your spinal cord. The true extent of spinal cord is ending at L1, L2 in case of adult and L3 in case of infant. Now here you can see this is a backbone opened up, dura is cut open, one vertebra is still there in place showing the vertebral foramen, and you are seeing the posterior view of it, and that's all your spinal cord extending. So in this case, if you see, so this is your C7 vertebra, cervical, and possibly this looks like your T12. I'm not sure, but this is what I am assuming. Yeah, it should be T12 because here somewhere it is L1 and L2. This is L1 and this is L2. Somewhere here the spinal cord is ending. And this is what is a bunch of your nerves, what you're seeing going down. And Above, if you see at this part, this is the part where your foramen magnum or C1 is there. So above this you can see this is the part of we call as a medulla oblongata, and below this everything is your spinal cord, right? Okay. So what is the length of this entire snake? So length is, it is 45 centimeter long in case of your adult male, slightly smaller, that is 42 centimeter in case of female. The easy way to remember this, as I always say, the easy way will be to remember this is one small scale, which is 15 centimeter, and plus one long scale, which is 30 centimeter. You put both of these together and then you will imagine that this is how big it is. Now weight, so it weighs around 30 grams, not very heavy. You don't feel the weight of your spinal cord in your backbone, thanks to the CSF present around it, which will take away the weight property. So you won't feel the weight, it is just the mass there. In the spinal cord, if you see, it is not uniformly thickened. It is some area it is more enlarged. So these are called as enlargement of the spinal cord. So in this case, you can make out here. So if I use a color here, so in this area, if you notice, in the neck region it is enlarged, we call cervical enlargement, and same thing here, the enlargement is starting somewhere here. So this one is what we call lumbar enlargement or lumbosacral enlargement. So there are two enlargements are there, cervical and lumbosacral. Cervical extend from your C4 to T2, and whereas your lumbar is extend segment, cervical extend C4 to T2 spinal segment, whereas your lumbar extend from your L2, L3, L4, L5, S1, S2, S3. So that is the extent. So the extent is, so it is, instead of being simple like this, so it start like this and then it will enlarge, then it is again narrow and again it is enlarging. So the upper enlargement is cervical enlargement, lower is your lumbosacral enlargement. So here the enlargement begins at your C4 and it is ending at your T2. Same thing here, it is beginning at your L2 and ending at your S3. So this is your enlargement of measurement. Fine. So now, so why there is a change here? I told you that in the adult it is ending at different level, and newborn it is ending at different level. So positional changes of your spinal cord will explain that. So in case of fetus, we are saying that it, when we are born, you can see even before we are born, so we can say intrauterine life. So that time, that is somewhere at the first trimester, at third month, you can notice your bone, so your vertebral column or your backbone, your vertebral column growth and your growth of your neural tube or nervous system, or I can call here as a spinal cord itself, both are same. So this is happening at till third month intrauterine life. This is intrauterine life before you are born. Now after that, what happens? There is a rapid growth in your vertebral column. So thereby the growth is, and there is a slow growth in your spinal cord. So thereby what happens is the bone grows faster than this. So at birth, the spinal cord is ending at your L3 vertebra, and further growth will happen in your body, and adult means puberty. So by that time what happens is the growth of your vertebral column is higher, and your spinal cord also picks up but not so much. So thereby it is now ending at, it is still not equal, so it is ending at your L1 vertebra. So this is due to difference in the growth of the bone and the nervous tissue, we have these changes are there. So you can see here in this picture, here both are ending at the same level, and so the spinal cord also giving the spinal nerve which are coming out at the respective foraminas, they will come out like this at the same level. But when the spinal cord lag behind in the race, so that time the wires of the spinal nerve which have come out, they came out. So what will happen is those wires will start elongating, and thereby they will become, see here you can see the wires are still coming out from the same place. If you notice this, sorry, if you notice in this picture, you can see here, this is your spinal nerve which is shown here, which is coming out, S1 spinal nerve coming out at S1 sacral vertebra. But now you see the nerve is still same, remains same, but you see the lower end of the spinal cord has gone to L2, and then you see again here is your again same S1 nerve is as it is, and the lower end is gone till your L1. So what has happened in this, in order to, because the nerve has already passed from the bony foramen, so the length of the nerve increases, but the cord decreases. So thereby not decreases, in fact we will say the growth is very slow. So this explains what are the positional changes. So thereby when we are doing the lumbar puncture, it is a procedure in which you put a needle to take out the CSF from the back. If you are doing in the adult, it should be always done below L1 level, whereas in case of your infant it should be done below L3 level. So you cannot do L1 level in case of infant, because if that is done you will injure the spinal cord. You can see, just imagine you're putting a needle, assuming that you can see in case here below L1 you put a needle here and you can take out the CSF. Same thing you try to do here, you will injure the spinal cord. So that is the reason it should be done. So in order to remember uniformly, always a lumbar puncture is done below L3 level, below L3 level, usually below L3 and L4 level is in both the cases. So this is the reason, anatomical basis, why lumbar puncture is done at this particular level. Now, what are the factors which are protecting your spinal cord? So yes, this is very simple and easy, and even that picture you can see it is protected by your bony cage, which is formed by your backbone or vertebral column. You can see there, and that is your outer very strong protection, and inside also you got a protection of your meninges, the coverings. Your dura, arachnoid and pia will be seeing in detail in a couple of slides. So these two, and other than that you pour a water in this now. So that is what is providing a hydrostatic cushion of CSF, cerebrospinal fluid. So fluid also protects, membrane is protecting and bone is protecting. It is something like your bone is like your army, navy and air force. So they are present at the border and they are doing the major form of defense. Then your membranes are, your meninges are like your what do you call, maybe like something like a police force. So which are there everywhere. The police stations are there, they are there, every part of it, which are protecting local law and order problem. And your CSF is more close, it is within, it is inside, because it is present between, it is very close to your spinal cord compared to your spinal meninges. So they are like something like a security guard of a building, a watchman. So who's doing more form of a protection. So thereby these are the factor which protect your cord, similar to the brain. Now, what are the factors which keep the spinal cord in position? Why spinal cord, the snake is not folding? Have you ever imagined why snake cannot fall back into the bottom of the vertebral column and it remains there? Or why it is not coming out from any of the side foraminas? So what are the factors which maintain its position? So one is the rostral. Rostral means the cranial. So cranially or superiorly it is continuous with the medulla oblongata. So it is something like the snake is continuous above with the medulla oblongata, that will ensure that it remains straight. And caudally, that lower end is fixed. So lower or inferior end of the spinal cord is fixed. That is, lower end is what we call a thread-like structure called phylum terminal. And you can see in this picture again, here, there are so many bunch of nerves are there. So this bunch of nerve is what we call cauda equina. We'll see this in detail in a couple of slides. So in the center you got phylum terminal, and on either side you got here a bunch of nerve called cauda equina. Both these are passing through the respective foraminas, and thereby they are also ensuring that the spinal cord remain fixed from the lower end. Upper end it is due to attachment to medulla, lower end because of these two structure called phylum terminal and cauda equina. You will know the details of these two structure in a couple of slides. So this upper fixing over, lower fixing over. What about the side now? So on the side, so it is on the lateral suspension, or the side support is provided by something called as a ligamentum denticulatum. Now ligamentum denticulatum are just like, they are your side, it is something like from your side of the spinal cord. There are some bands are coming out, and these bands are going and they are attaching to the dura mater. So these bands are called, they look like a teeth, that is why I call it a denticulatum, and they are ligament like structure. So ligamentum denticulatum. Details also we'll see about all these three: phylum terminal, cauda equina, ligamentum denticulatum. All three are 3 and 5 marker questions, so I'll be doing in detail in a while. So that is your support what we have. That's the reason why the spinal cord is in position. Now we'll start with the coverings of the spinal cord. They are called as the meninges. So meninges are, 3 meninges are there. The outer meninges is what we call as a dura mater. So the word dura comes from the word durable in English. This is how the Latin word durable came to English, and that's how we use the word durable, means tough. So dura means tough mother. It is also called an alternative name called pachymeninges. So this anatomy guys might be aware, because I have taken this part in the head and neck part of it. So pachymeninges is nothing but a dura. Dura means tough, and the word matter if I add. So what is the meaning of matter? Matter means, the word matter comes from the word mother. Mother means mother. So why mother? Because mother is meant for protection. So mother protect the child. That is the reason, protection. So that's dura is tough, madar is mother. So it's a tough mother which is protecting. So here in this case, this one is your dura mater which is cut open here, and this is your closed dura mater. Please notice the dura mater is not only present on the spinal cord, but it is also going along your spinal nerve. So that is also the extension of dura mater along your spinal nerves. Okay. Now you got the second layer and third layer. The second layer is what? Arachnoid. Arachnoid means spider web like, and pia means thin. So these are your spider web like mother, and here you can see this one is your arachnoid matter, and here whichever the transparent thing which is lying on your spinal cord, that one is your pia mater. So that is your three coverings are there. Now you can compare the dura mater with the blazer. So dura can be compared with the blazer, what we are wearing outside, blazer or a jacket or a sweater. Arachnoid can be compared with the shirt, and pia can be compared with the undergarment like banian or something. So thereby you see the blazer is much more stronger than the undergarment, that banian and all. So that is the reason. So these are all things are protecting, and our skin is something like we are the spinal cord. So that way the protection has been provided. And between your shirt and the undergarment, that is shirt and the banian, there is a water column is there, and which we call as a CSF, which is providing the protection. Okay. So both arachnoid and the pia mater together are called as leptomeninges. So leptomeninges have been asked in the MGR University as a 2 marker. So if you know leptomeninges means arachnoid and pia, you will be able to write. But if you don't know what is lepto, then you will not be. Lepto means literally means thin. So they are together, they are your thin coverings. So thin mother or thin matter. That is what is the meaning. So first we start with the dura. So dura mater, you know, in case of brain, the dura mater is made up of 2 layers. The outer layer, which is attached to the inner surface of the wall, is what we call as your cranial dura, or the dura of your brain, is made up of 2 layers. The outer is called as your endosteal dura, which is the one which is attached to your endosteal layer, which is attached to your inner surface of your bone. An inner layer is attached to your arachnoid matter, so it is called as a meningeal layer. So there are 2 layers are there. So this meningeal layer of your cranial dura, this is your brain, and this one is your meningeal layer, and this is your foramen magnum, and this one is your endosteal layer, and this one is your skull. So you can see here at the foramen magnum, it is a meningeal layer which is coming down and become the dura mater of your spinal cord, right? So this is your meningeal layer. So thereby the dura mater of your spinal cord is a meningeal layer of your cranial dura which continue down. So meningeal layer of your cranial dura, so meningeal dura of your pia mater. So your dura mater of your spinal cord is a meningeal dura of your cranium which continues as a spinal dura. So extent is, it extends from again foramen magnum to your second sacral vertebra. So all the way from your foramen magnum, somewhere here you can notice here, if I use here if you notice, see down in the picture, so there you can see, so this one is your dura, this is very thick, you can see that's your dura mater here. The same dura is coming down with from your foramen magnum, and it is becoming your dura of your spinal cord. So this is a dura mater of spinal cord, right? Same thing goes down all the way. So that's here, and the extent is, it is, we can say that it is starting from your foramen magnum. In this case, this one is your foramen magnum. It starts from foramen magnum, and below it goes and goes all the way down to your, here you can see it is going all the way down to your sacrum. Somewhere in the sacrum it is continuing. Here we cannot make out, the clarity is not very good, but it ends into the sacrum, and sacrum also it is to the second sacral vertebra. So this is the covering till your second sacral vertebra. That means your S3, S4, S5, that is your lower part of your lower 2/3 of your sacral canal is without dura. So there is no dura there, and beyond this, so that means after this part, so it is continuing as the dura continuation. Beyond S2 vertebra, it continues as the dura of your phylum terminal. That means spinal cord is not having, beyond that it is just covering your dura of your phylum terminal. Phylum terminal, maybe the picture will come, that time you'll understand. Now epidural space is a space between the bone and the dura. So the vertebral column and your dura. So outside this you have the vertebral column. So the space between these two is what is called as a your epidural space. It is also called as a extradural space. So extradural space is the space between your dura mater and the vertebral column. Now there will be a space between again the dura and your second layer called arachnoid, and that space is what we call as a subarachnoid space. So subarachnoid space and epidural and extradural spaces are the spaces above and beneath the dura mater. So here in this case, so the space what we are seeing here now, because this is your dura mater and this is your arachnoid mater, so this space I can call as a subdural space, and here outside this space will be your epidural space. Okay. Now this is a picture again from Vishram Singh. This is very simple. So if you take a cut section through this, take a blade and cut it right through it, like through and through if you cut, or here if it is close, if you cut like this, then you will get something like this, a cucumber cut of your spinal cord. Here you can see the coverings are very simple from outside to inside. The outside outermost is your vertebral canal, so that is your backbone we can say, and deeper to that you got your dura mater, which is here the dura mater green color. In between them whatever is present, that space where you are seeing that blue structure, this is what we call epidural space. Now the epidural space is important space because it is a space which is clinically used. Now this epidural space is a space which contains some structure. So I would like to add some details on this before we move further. Can be asked in the exam. So epidural space, it is also called as a extradural space. Extra means outside, so extradural space is the other name. Now here location you already know, so it is between your vertebral canal and spinal dura. This space is absent in case of your cranial dura, so you don't have any epidural space, potential space, though epidural hemorrhage may happen there, but that is only when hemorrhage happens the space is created, but in reality there is no space. But in case of spinal cord there is a physical space which is present. This epidural space location. Next is the content of the space. So what do we have in this space? There are so many things are there in this space. One is your loose areolar tissue, and those blue structure what you were seeing, they are your vertebral plexus, so internal vertebral venous plexus. And you will also see that here you can notice that these nerves, these all the nerves whatever you are seeing, they have to actually pass through this. So they are coming out here, means these are the structure which are piercing and coming out. So thereby you also have these structures. So thereby you can call them as a roots of your spinal nerves. All that 31 pair of spinal nerves, they have to pass through it. So roots of spinal nerves. Then in addition to this, the spinal nerve or spinal cord is supplied by your arteries. So thereby again, by end of the topic you will understand this part. They are called as the spinal branches of regional artery. You will know what I am talking later on, regional arteries. And then further you will also have some nerves which are actually going back. So there are nerves which will go back from the spinal nerve. So those nerves, suppose in this diagram there are some nerves are there which will actually go back. So they are coming out like this. Some nerve will go back into it. So those nerves which are going back, they are called as a recurrent nerves. So they also again will pierce back second time. So thereby you call this as a recurrent. And why they are going back? Because they are the sensory nerve which has to supply your meninges. So they are called as a recurrent spinal meninges, recurrent meningeal branches. So recurrent meningeal branches, they are the branches of your spinal nerves. And to protect all of the structure between the bone and the dura, you should have some cushioning. So, and you have the fat, but having a solid fat will cause the compression. So the fat is in the form of a jelly-like, so semi-fluid state of fat. So these are all the structures what we have in this space. Now what is the applied aspect of this? So what is the application, clinical application of this? So the space is used to numb. So here we give an anesthesia that is called as a epidural anesthesia. Epidural anesthesia is given in this space. And why we give this in this space? Because the spinal nerves are piercing the dura, and you give the solution there, it will cause numbness. So it will numb. It will numb the spinal nerve. Numb means it will actually block the spinal your nerve transmission. It numb the spinal nerves which are traversing the space. So wherever you are giving this, if you give in the upper part, it is doing lower part. So whichever level you are giving, from that point onward, the below that particular level. Suppose if I am giving somewhere at C3, C4. So from C4 onward till the end of it, all the spinal nerve which are traversing the space, they all will get anesthetized, and they all will have numbness, and the area supplied by them also will become numb. And usually where it is given, this is given in the cases where this approach is used to, mainly in the cancer patient. So it is to relieve of pain it is a palliative treatment, cancer patients, where your local or your analgesic, your painkiller, when the painkillers, the normal painkillers what we are giving by oral or even by injections are not effective, where painkillers are not effective. In that case, we have to take a little deeper approach. So that is, and this epidural anesthesia, we know that your dura is ending at S2 level. So beyond that you don't have a dura. But beyond that you definitely have a space, because the space is between the bone and the dura. So there is a space. So we can say the lower limit of the dura mater, especially at S2 level and all. So here we can say the space is very wide. So you can say beyond the lower limit of the dura mater, below this, I can say below S2 level, your epidural space. So this is the space I can say in the lower 2/3 of sacral canal. You may, anatomy guys should follow what I am saying. Sacral canal. So this is what I am saying. The lower 2/3 of the sacral canal. So that space I am calling epidural space, or I can call this is the sacral epidural space. And sacral epidural space is the space which is in the lower 2/3 of the sacral canal. Now that space is, what happened to this space? Now this space is wide because you don't have the dura there, no arachnoid, nothing. So the space is quite wide. So in this space, and this space extend all the way from your lower 2/3 of the sacral canal to all the way to hiatus, sacral hiatus, which is the lower opening of your sacral canal. Sacral hiatus was asked as an exam question. So we can, at the sacral hiatus, anesthetic can be given, an anesthetic agent or anesthetic drug can be given, is given through this. This is what we usually do in case of your labor also. So this is again epidural sacral anesthesia, or also called, since you are giving it the lowermost end, this is also called as a caudal anesthesia. I told you about when I was doing the perineum and pelvis. So caudal anesthesia is given in this space. Yeah, so this is about your epidural, right? So now there is a next layer is there in this. The next layer is your black line which is shown, and that is your arachnoid matter. So we have to look at the arachnoid matter now. So arachnoid matter is not much complicated here. Before we go there are some differences are there between your dura mater of the brain. So this is your spinal, this is your brain dura. So this is the dura of your brain. So brain dura versus spinal dura mater. What are the differences are there? So again, very important to know for the anatomy guys. So they both are not same. Here it is a single layer, whereas here it is a double layer. And the double layer is your inner meningeal layer and your outer endosteal layer. I told about this part, and here it is only your meningeal layer what we have, and they do not form any folds. So what do you understand by the dural fold? The dural fold, you know, they are your falx cerebri between two cerebral hemisphere, falx cerebelli between two cerebellar hemisphere, tentorium cerebelli between your occipital lobe of cerebrum and your cerebellar hemisphere, cerebellar lobes, and diaphragm muscle separating or forming the roof of your pituitary fossa. So these are all the different folds we have learned in the head and neck. So these folds are present here because there is a double layer. Here it is a single layer, so we have no chance of forming a fold. Now this is quite important, that there is a space is present, epidural space is present, where the epidural anesthesia is given. Here you don't have as such this space. Space is absent, but the space can be created if there is any hemorrhage of the dura mater and the blood from the middle meningeal vessel can come, and so thereby epidural hemorrhage is possible even without having the space, because the space get created. So these are the differences. Now here this is again a picture which is showing here all the spaces in real form. So you can see again here. So here we have, this one is your anterior, because here is a vertebral body, and this is the spinous process, this is a posterior, and these are your lateral transverse process are there. So you can see here all this, these are your spinal nerves which are coming out from either ends, right and left, and the coverings are there again here, go from the outermost dura mater, then arachnoid mater, second layer, and the third layer innermost close to the spinal cord is your pia mater. Now please concentrate on the spaces now between your bone and your dura, where you see the fat is there on this side. There is a fat on this part of it, if I use a different color, and here it is shown empty. So this is the part. So that is your epidural space here. That's your epidural space what we have. In the same way we have another space between your arachnoid and the pia. So where you are seeing this one, this is the space where I am running the orange color now. So that space is called as a subarachnoid space. So these are the spaces. Now look at the epidural space. Here we can only see, in this case here you can see there is a fat in the front. I'll use a black color maybe. So you see there is a fat here in the front, and there's a fat here in the front. In addition to this, you will have the vertebral venous plexus and other content whatever, and even you see the same space is continuing on this side. See, there's a continuity of the space here. So the space here, it is pierced by, here you can see it is pierced by your nerves. So here the nerves are piercing, and you also notice your spinal nerves are covered by your, see, this is what is the covering of your dura. So thereby the space is extending even to your spinal nerves also, right? This I was telling about the anesthesia, epidural anesthesia. So where the needle given, so it is, you can see it is, we are passing through a catheter, we leave the catheter inside, then we connect it to a machine. So this is a machine through which you can actually regulate the flow of the your analgesic or painkiller. So this is how in the terminally ill or long surgical patient we will put this one. This is how it looks like, epidural anesthesia. And wherever you give, this is what is the area of numbness below the point of wherever you give. So especially during your pregnancy and all, you give this as a spinal anesthesia. And the person who needs epidural is a pregnant lady. Here it is a joke. It is not that the male is delivering, but it's just a role reversal. But during pregnancy, parturition, delivery, this is given, right? Now next layer is your meningeal layer. So meningeal layer we got here, this is what we call as a arachnoid matter. So arachnoid matter, word come from the word arachnoid, means spider web like. So this covering is thin. This is thin and transparent. You can see here in this case, this was your dura, which is thicker one, and you can see a transparent, semi-transparent covering is your arachnoid matter, and it is not as thick. If you see the thickness here, this part of it, and see the thinness here, this part of it, so they are not very thick, they are thin covering and transparent. And it also has a space both outside and deeper to it, the space which is separating from the dura. So thereby I can say this space. So this space where I am putting the star is your subdural space, which separate your dura and your arachnoid. And I put the hash now. So this is the space which is separating your dura and the pia. So this is what we call as a arachnoid and the pia, we call it as subarachnoid space. Now the extent of this also is, above upper extent is same, it is from the foramen magnum. Lower extent is also same as your dura, that is to the second sacral vertebra. So beyond second sacral vertebra you don't have this covering, so it is ending there. So that is the lower end of your, or lower extent of your spinal arachnoid. Now that's it, that's about it, nothing big deal about it. It is delicate covering. Now next we move to the pia mater. pia mater is, pia means thin, mater means mother, and it is also extending all the way from the same thing, that is from your foramen magnum above. Lower down, it is not, it is reaching your conus medullaris, which is the apex of your spinal cord, and from that point onward. So if this is what is your apex of your spinal cord, which we call as a conus medullaris, and this is the covering which is lying very close to it. So the right and left pia mater comes at this part, and so right and left part which is coming at this part, instead of instead of ending it here just like they have ended, it could have ended at L1 or L2 level, but what is happening is it is going beyond, comes and meet at the apex of your conus medullaris, and at the apex it is forming a single thread like structure, and now you call this as a phylum terminal. So phylum terminal is the continuation of your pia mater beyond the lower end of the lower extent of your spinal cord. So this continue or extend as a final terminal, and where phylum terminal will go, it will go all the way till your coccyx vertebra. So this is where it is ending. So thereby you can say among all the three covering, the lowest covering is your pia mater in the form of phylum terminal. We'll see the phylum terminal in detail also, because it itself is a 3-marker question. Now please notice here, it is the one where you have the vessels lie along this layer. This is a very important layer. So here you got the vessels all along. Here also in the picture also you can see your blood vessels are there. So here this one, here you see this one is here, or you can see this is the covering, that's your arachnoid here, and that's your pia there, and you can see the blood vessels are lying in a very close intimate contact. The pia mater is important. Here this is again just to show you the spaces. So let us look at your arachnoid mater. So that's your arachnoid mater, and then there is a pia mater. So that orange one is your pia mater, and black is your arachnoid mater. In between them you got a space, the largest space I must say, and that space is your subarachnoid space. Now in this space you can see there are so many, it's a space is so wide that it is filled with so many other structures. So I will talk about the content of the space. So I can now, I think next is, you can see here, here it is already shown again. So this one is your epidural space with the fat and the veins, internal vertebral venous plexus. You also can see your dura mater, there's arachnoid mater shown there, and that's your subarachnoid space, and there is a pia mater enclosing the nervous tissue, and this is filled with your CSF. So CSF is the one which is present in this particular space. Not only CSF, but also you can see in this picture, if you see, it is also filled with the blood vessels. So thereby, so if you ask me what is the content of, so this one, if it is subarachnoid space, can also be asked as a question in your exam. So subarachnoid space, these two spaces are important from exam point of view. So subarachnoid space is a space which is of uniform size, at least up to, it is of uniform size. So first you can say about the location. Location is between your arachnoid mater and the pia mater of spinal cord. If I am talking about subarachnoid space of spinal cord, subarachnoid space is also there in your case of your brain also, that we call the cranial subarachnoid. So cranial subarachnoid space is continuous with the spinal subarachnoid space at the level of foramen magnum. So this is of uniform in diameter. So uniform in size, I must say, uniform in size till your conus medullaris, that is your lower end of your spinal cord. So it is uniform in size till the conus medullaris, and beyond this what happens? Beyond this the size increases. So beyond conus medullaris, size of subarachnoid space increases, and this increase is what we call as cistern. So cistern is a dilated subarachnoid space. So what is a cistern means? It is a dilated subarachnoid space. So when space is dilated, what will happen? A cistern will contain more CSF. So there is a more amount of CSF present in your cistern. So since the cistern and this conus medullaris is ending where? At your L1 and L2 in adult. So thereby below this is what we are talking. So thereby this dilated space is present in the lumbar region. So thereby it is technically called as a lumbar cistern. So lumbar cistern is a cistern, a dilated arachnoid, subarachnoid space present below your L1, L2 level. Now this space, lumbar cistern, extend up to your second sacral vertebra. So thereby we can say it is starting almost at L2 or L1, can go till your S2 vertebra, because after that you don't have the arachnoid matter. So this is the extent of this space, quite a big space. So thereby this is what is your lumbar cistern extent. Now what do we have? So in this, so what are the structure which are present inside this, the content of the space? So content is, the very important content is cerebrospinal fluid is present, because of which you don't feel the weight, provide the buoyancy. In addition to this, you have bunch of or collection of bunch of nerve roots are present here. Bunch of nerve roots which are surrounding your thread-like central structure called as a phylum terminal. So phylum terminal goes all the way till your coccygeal vertebra. So thereby phylum terminal is present along with a bunch of nerve roots, and you also have in this space your of course the blood vessels, are also located here. Blood vessels, yeah, these are the structures what we have here. Now when we say bunch of nerve root, especially here we have something what we call as a nerve root which is ventral and dorsal rami starting from your L2, L3, L4 and L5, S1, S2, S3, S4 and S5, and your coccygeal. So these are the nerves. So if you count, these are 4, this is 5 and this is 1. So total there are 10 on right side and 10 on left side. So thereby you can say 10 pairs of ventral and dorsal rami are the nerve. So it is forming a bunch here. And this bunch, which is made up of the 10 pairs of nerve, resemble a tail of a horse in this space. Tail of a horse. That is why it is called as a cauda equina. So cauda equina, cauda is a tail, equina is a horse. So what do we have? So the content is, you can say bunch of nerve, or another word I can say, the content is a cauda equina. So cauda equina is a bunch of spinal nerve roots located in your lumbar system, formed by ventral and dorsal rami of L2 to L5, S1 to S5 and coccygeal nerve. So this is what we have here. And what is the this is the content. Now what is the significance or clinical importance of this space? So since you have here CSF is present here, so we will do definitely. So lumbar cistern is approach. So this lumbar cistern is approach for doing your CSF puncture. So CSF or lumbar puncture is what is done. So this itself can be a 3-marker question. Cistern, CSF puncture or lumbar puncture, it is done in the lumbar cistern or subarachnoid space. And why it is done? To collect a CSF sample. Small amount of CSF sample is collected here for investigation in laboratory to know if there is anything wrong with your brain or spinal cord, because this is the fluid which is washing or moving around the brain and spinal cord. So if any bacteria infection is there, so those waste metabolites or their toxic or their antibodies, whatever is there, they will be released into this. So it will give fairly a idea what is wrong with it. So this is one use of it. So what is the use? You take the CSF sample from it. The second use of this is the same space can be used to give your anesthesia. Your spinal anesthesia is given here. So spinal anesthesia. So epidural is outside your dura and your between your bone, but spinal anesthesia is between your arachnoid and pia mater. So spinal anesthesia is given here, and usually it is between your L3 and L4 only, because above that you may damage the spinal cord. So what happens is the spinal anesthetic drug, it will dissolve. It dissolves in CSF and thereby bring about its required action. So thereby it will bring about the numbness depending on which nerves are passing there. So usually you can see that all your cauda equina nerve are the one which are in this area. So all the areas supplied by your cauda equina. So that means you already know your lumbar, so thereby we are looking at lower limb, and your sacral nerves, so we are looking at your pelvis and perineum, coccygeal also. So thereby there will be area of numbness. So there will be area of numbness in your lower limb and pelvis and perineum. So that is a sensory part of it. Motor part of it will be your paralysis of your muscles of your lower limb and your pelvis and perineum. So the numbness what develops in your this chorda equina is what we call as a saddle anesthesia. It's just like when you're sitting on a chair, so whatever part of your bum's coming in contact with the chair, there will be numbness developing that area. Sometimes it so happens that when we sit for very long time on a hard surface continuously, and when you get up you feel that because of the continuous compression in that area, so some part of your bum's inferior aspect will feel little different. So that is exactly the numbness which has been felt when the anesthesia is given. And same thing if your chorda equina is damaged because of any reason, any fracture or any damage happen in this area, there will be involvement of these nerve root, and the outcome is what we call as chorda equina syndrome. So chorda equina syndrome happened due to damage or involvement of the chorda equina nerve roots, and the symptom will be the patient will have the weakness in the lower limb muscle and the pelvis and perineum muscle, and there will be loss of sensation in the pelvis and perineum and lower limb, that we call saddle anesthesia. So because of this the patient cannot walk, walking is affected, and because of this the bladder, which is your urinary bladder, and bowel, which is your large intestine, especially your anal canal, so the external anal sphincter and external urethral sphincters are lost, so thereby bladder and bowel control is lost. So this is what is your chorda equina syndrome. So chorda equina syndrome, what happens is, so chorda equina syndrome, it can happen because of anything which damages those nerves, tumor, hemorrhage, fracture, accident, infection, anything. So there will be motor symptom will be there. So motor symptom will be, there will be lower limb weakness to begin with, and later stage is paralysis of muscles. So thereby they cannot move or walk. And there will be weakness, pelvis and perineum, weakness and paralysis of muscle. Now because of this weakness, person cannot, inability to walk, so walking to walk, and because of this your anal sphincters and urethral sphincters are gone. So anal, external anal sphincter and your external urethral sphincter, which are supplying your anal orifice and the urethral orifice, so thereby there will be loss of control. So we call that as a anal incontinence, that is unable to hold the feces, and urinary incontinence as dribbling of the urine, unable to hold the urine. Incontinence. Continence is to contain, incontinence is inability to do. Incontinence. So this is motor. So what will be your sensory symptoms? Sensory symptoms will be same. So in both lower limb and your pelvis and perineum, there will be loss of sensation to touch, pressure, pain, temperature, whatever is there, especially in pelvis and perineum. Lower limb it may not be, because you still have lower limb also, yes, because depending on, because we are looking at L2 to S4, so thereby lumbosacral, that is your even sciatic nerve is gone, so thereby definitely there is a loss of sensation in lower limb. So here also you have a loss of sensation in your pelvis perineum, and we use the word as a saddle. Saddle is a seat, on we keep it on a horse, leather seat. So when you sit on that, whatever the part comes in contact with that, so that area you will have the loss of sensation. So that is why it is also called saddle anesthesia. Saddle anesthesia. So this is about your subarachnoid space, very important space. So here this is how the CSF is withdrawn, and remember the CSF is withdrawn always below. In case of your infant it should be below L3, in case of adult it should be below L1. So ideally they do it as a uniform L3 and L4 at that level, or below L3 and L4 always. So usually they will at L4 level they do it. So usually they do it at L4 level uniformly, where you draw a line which is meeting your posterior superior iliac spine. So where is it? Here, somewhere here, not the posterior superior, the highest point of the iliac spine is what we call iliac crest. So a line is drawn from there which passes through your L4 spine. So this is where the L4 spine it is passing, and here you have the highest point of your iliac crest. So here highest point both right and left, and this is the point where immediately above or immediately below this is where your it is done, and always the patient is asked to flex. In the supine position, the patient is asked to bend forward. So by bending forward, the space between the two vertebrae opens up, and also your spinal cord slightly moves up. So this will improve the chances that there will not be any injury to the, any slightest injury also may not happen to this. There is a chance that sometime what happens when we are doing this, that there is a possibility that the needle may pierce any one of the cauda equina nerve. So if that happens, a slight pain may happen, but that particular nerve will regenerate back and the patient may not have any neurological symptom. So you can see the needle is going. So if you see the anatomy point of view here, so what are the things? So it piercing, we are piercing the skin, superficial fascia with the fat and all. Then there are muscles what we have, all the back muscles we got there, and then we got here, this is your spinous process of L3 and this is spinous process of L4. So between these, so before you have here, there is a supraspinous ligament is there. Now this is your infraspinous ligament, this one is your supraspinous. So we are piercing supraspinous ligament, then we are piercing your interspinous ligament. This is the interspinous, supraspinous and interspinous, and then you can see we are going further deep inside. So after piercing the ligament, we are going now into your dura mater. So that's your dura here. So this one is your dura. So dura is, here is your dura, and this one is your subdural space where I am putting the star, and this one is your arachnoid matter. So this line is your arachnoid, which I am drawing now, that is your arachnoid. Then this empty space what you are seeing where I am putting hash now, that is a subarachnoid space. And this bunch of nerve what you are seeing is your cauda equina. And here you will have your CSF. CSF will be present in this space. So thereby, when you are passing through this, when you are taking this journey, there is a feeling of, as soon as you pierce the arachnoid matter and you are entering the subarachnoid space, the feeling of giving away. That means suddenly the resistant journey will become free. That is how you will know that you have entered the right place, and you can withdraw. Very rarely it may even touch the nerve also, but every time whenever you push anything inside this, the fluid will cause some movement. That movement will disturb the spinal nerve. So it is something like you shooting an arrow by being in a swimming pool and trying to hit some rope which is freely moving in the water. So the chances are very rare that you may hit, but once in 1,000 also if you hit also, there will not be any neurological deficit and the person will have a regeneration of whatever damage which has happened on its own. So this is about your site of needle insertion for a lumbar puncture, and you can see how the anatomical knowledge is helping you to go this. If you do this below or above your L1 level, so you are directly going and you are injecting, you will get CSF still there, but you are injecting or you are damaging the spinal cord. So usually in the cadavers, during your autopsy or post-mortem, so that time they usually can do it anyway, because anyways the person is dead and they don't it doesn't matter much, so they will not look for it, but it is always better to do it in the lumbar cistern, because it is a big pool of CSF and it is always easy to get more amount of fluid from that area, right? Okay. So here you can see in the living person, here we have the patient is lying on his side in this particular case. The head is on the left side of the picture, so thereby something here is your head, this side, the lying here on the, and then after giving a local anesthetic, lumbar puncture needle is introduced through the third and fourth lumbar vertebra interspace, and a stylet is withdrawn and a drop of CSF should appear, and a manometer. You see this one is a manometer. Manometer is important to know how much pressure we are doing. So the pressure important is there, because if it is pressure is not maintained, more amount of fluid may drop in and it may cause the herniation of the brain in the foramen magnum. So there is a three-way valve is there. So one is for the manometer one, another one is down here, you can see the drop is here, so it is down for your outlet, and one more is here, you can see this one, not this one, which is going inside this. So this is also provide the air, also air entry. So CSF pressure measurement is important for the collection of your fluid. Entering pressure can be determined when the needle is inserted into the subarachnoid. So the pressure changes, and that is how we know that we have entered into the right space. And the lumbar puncture is technically also called as a spinal tap. So lumbar puncture is one name, spinal tap is another name, and what name could be your lumbar puncture, spinal tap? Yeah, these are the two names what we have. Spinal tap is a clinical name. We are tapping the fluid from the spinal region, that is why it is called spinal tap. So this is normally a normal good fluid, or a clear fluid in you and me should be very clear. It should be crystal clear like a water. But when you have infection or something, a cloudy specimen may be there, just like it is shown here. You can see it is very white or milky white, which indicate pus. That means there is a increased number of WBC in this. So this could be, again, the most common cause is your meningitis, inflammation of meninges in the brain. So again, even a blood can also be there in this, which could be indication of a subarachnoid hemorrhage. So a subarachnoid hemorrhage, because the blood vessels like circle of Willis is present in the same subarachnoid space, so thereby if the bleeding is happening, you may find even the red color inside this. So that red spot, if you have inside this, which can also indicate that there is a hemorrhage, subarachnoid hemorrhage. Now modification of pyometer. So we are done with the dura, we are done with the arachnoid, and we have done with the epidural space and the subarachnoid space. So sub space, one more space was there here, which I didn't mention because this is not clinically important. Here we have another space, now which we call here, this is a space, very small narrow space if you notice. So this space is what we call as a subdural space. Now subdural space is clinically not important. Here you will have a venous plexus is present here. So venous plexus. So that's about your, okay, now we go to the pyometer. So pyometer is very important in case of spinal cord. Many questions have been asked in this. So we know pyometer is the thinnest covering which is lying closest to the spinal cord. So you can see, so in this case here, this one is here, covering will be here, this is your pyometer here. And this pyometer shows a lot of modification here. So these modifications are not seen elsewhere, even in your pyometer of your brain. So these modifications are limited to your spinal cord. Just like your durameter is very well modified in case of your cranium or brain, they form your dural fold, dural venous sinuses, so on and so forth. But here we don't have those modification. But the pyometer is very simple in case of your brain, but here it is modified. So modification of durameter, like your dural fold, falx cerebri, falx cerebelli, tentorium cerebelli, and diaphragm ocelli are important question from university point of view, including your dural venous sinuses. The same way when it comes to pyometer modification in your spinal cord, they are important from the university point of view. So it shows 4 modification of your pyometer in your spinal cord. The first modification is what we call as your phylum terminal. The second one is called subarachnoid septum. Third is called linea splendens. And the fourth one, so fourth question has been asked. This is your ligamentum denticulatum, has been asked in the exam twice as a 3-marker question. So the other phylum terminal is a very often question. This has been asked as a 3 and 5 marker many times. Ligamentum denticulatum is a 3-marker question. At least it has been asked twice in the university exam. Other 2 they have not asked. So let us see in this case. So here in this case, if you are seeing, if I have to show you in this case, so this red structure what you are seeing, which is passing through this one below the conus medullaris, this red structure is your phylum terminal. So that is your phylum terminal here in this picture, and this needle is showing the lumbar puncture, how it is done in the same space. So in this particular case, we are not seeing the cauda equina. The nerves have been removed here just to show the clarity of the phylum terminal. And next is, if you want to see your, in this case, if you want to see on either side, if I put, can you see there is one septum is there on this, this is too thick. So if you see, there is one septum on this side and this side. That small septum what you are seeing between your ventral and dorsal root. So that septum is what we call ligamentum denticulatum. We'll see the detail later. And this is one part which is again providing the lateral support. Now this is your posterior end because it's a spinous process. This is your anterior end because that is a vertebral body. Here you see there are again in this area, if you notice, you can notice here in this part, these ends are there, these structures what you are seeing which are present here. Now these structures are called this one. This is the one what we are present which is behind. So this is the posterior area we are looking. So posteriorly these are called as a subarachnoid septum. So these are your subarachnoid septum. Again the modification of your pia mater posteriorly. Same thing if you come anteriorly. Here in this area there will be a thickening part will be there, which is again not very clear in this picture. So that thickened portion which is present in the anterior part is what we call as a your linea splendens. I have a photo for all of this individually, but in this picture I am showing you. So let us see this one. In this picture, here you can see there are here tooth-like structures which are there here on either side. Can you see this one? On both sides. So these tooth-like structure are called ligamentum denticulatum. Denticulatum because like a tooth. Okay. We'll start with the phylum terminal. Remember, it is an important 5-marker or 3-marker question. Now we know phylum terminal, we have already been introduced to this word. So phylum means actually a thread. So in government offices, old offices and all, nowadays it is being replaced, but still some villages it is there. Or even when I used to write exams, so there were no staplers and all. So that time they will give us one thread and we make a hole and we used to pass this thread through all the answer sheets and label it. So they used to call this thread as a file tech. So it is that phylum word come from the word file. File means actually nothing but a thread. So here, and it is a terminal part. It is present in the terminal part of the spinal cord. That is why it is called as a phylum terminal. Now, phylum terminal is, here the first word should be, it is a modification. So what is it? It is a modification of pia mater. This is important. It's a modification of pia mater seen at the lower end of the spinal cord. Which part lower? At L1 lower end of the spinal cord, which is nothing but forming a cone at conus medullaris. So it is starting from, I can say it is starting from the apex of, so it starts from the apex of conus medullaris at your L1 or L1 L2 vertebra. That is what is the beginning of it. And what is it? It is a continuation of the pia mater from the spinal cord. So beneath the conus medullaris, the spinal pia mater continues as your phylum terminal. So length of it, if you measure this length, entire length from this end all the way till this end, here you can see this, the entire thing, if you measure, the length is coming to 20 centimeter. Almost you can say 2/3 of your, it is slightly bigger than your small scale, but so 2/3 of your large scale is what we can call as a this here phylum terminal. Almost you can say like more or less like a forearm length. It is that big. So it is again divided into two parts. The first initial part which is lying within your dural sac. You can see this is what is your dura mater with the arachnoid mater. You can see here this is your dura mater. We said the dura mater will go till your S2 vertebra. Remember the dura is extending till here and arachnoid also is extending till here. So beyond that you don't have the dura. So the part of the phylum terminal which is lying within this dural sac is what we call phylum terminal internum. And this is amounting to your 15 centimeter. So it is around your 15 centimeter in length. And the part which is lying outside this dural sac, which is 5 centimeter, and this is what we call phylum terminal externum or externa. So it is again a 5 centimeter long, it is outside. And this is the part which is extending from. So what will be the extent of your phylum terminal internum? So phylum terminal internum extends from here L1, L2, that is from the tip of the conus medullaris till your second sacral vertebra, till your lower end of your dural sac. So that is the extent of your phylum terminal internum. Externum will be from your S2 vertebra all the way till your coccyx vertebra. So that is the end of it. Dorsal surface of coccyx, because this is the anterior, this is posterior. You are seeing it is ending at the posterior aspect of your coccyx vertebra. So that is your phylum terminal. So this ends at your coccyx vertebra. Your point where internum and externum are different, so that is your S2. Above S2 is your internum, below S2 is your externum. So what is the function of it? If you remember in one of the slide here, I told you the function I mentioned in case of here when I was talking about the supports of your spinal cord. There I mentioned that spinal cord has got cranial support, caudal support, and here I mentioned, you see here, remember this part of it, and here I told you the caudal fixation is provided by your phylum terminal along with your cauda equina. So phylum terminal along with the cauda equina. So here in this case, if we go and you see here in this part, the centralmost portion here, the centralmost part here is centralmost thread-like structure which will continue down all the way. You see here it is ending at the coccyx vertebra. Did you notice it is ending at the coccyx? So that portion is your phylum terminal internum. In this also you can see this one is your dural sac. Can you see this is a dural sac which is ending here? So this one is your S1 and this is your S2 vertebra. So it is ending at your S2 vertebra. So from this point, that is from the apex, here you can see there is the apex of your conus medullaris till this part, this one is what we call as a phylum terminal internum. The part beneath it till your coccyx vertebra on the dorsal surface, this portion is what we call phylum terminal externum. Now what is the function of it? It provides your caudal support, caudal fixation. Your spinal cord is provided caudal support to the spinal cord. So in this point you have to mention, because here I think it is not been written here because it has already been explained there. So the function of this is, so function you can write here, function as provides caudal. Caudal means inferior. If you don't remember caudal, write inferior. Caudal support to spinal cord. It is something like if spinal cord is like a kite, a phylum terminal is like a thread, and your coccyx is your hand which you are holding, or a stone or a pipe where you have tied up this. So thereby, as long as it is tied up, your kite in the sky is moving all along it, and the sky is your CSF all around. That's how it is providing the support. That's an analogy. Now the second modification, remember we are done with the phylum terminal. Next is your subarachnoid septum. The subarachnoid septum, I told you it is present in the posterior aspect. Now if you take a spinal cord here, and I told you here you can see spinal cord and posterior aspect of it if you notice, and here you can see this one is your pia mater on either side, right? And this is called posterior. So here this one is your spinous process, so this is your posterior end of it. So here in the center, if you can notice, if I use a different color, you see there is some extension which is coming out from the back, these extensions, and I'll rub it now so that you see those extension clearly. Now these extension which you are seeing now are what we call as your subarachnoid. Why subarachnoid? Because it is starting from the subarachnoid, it is running in the subarachnoid space, so thereby you can call it as a subarachnoid septum, right? So it is subarachnoid septum. It is present in the mid sagittal fenestrated pile septum, which is connecting your dorsal surface of the spinal cord with the arachnoid matter. So here you can see the connection. Here also you can see in this picture from your Vishram Singh, you can notice your subarachnoid septum is here, and this is the area which is shown. So thereby the connection, it is connecting here. This is what we call dorsal, because here is the anterior, that is posterior or dorsal surface of your spinal cord is connected to your arachnoid matter via the modification of pia mater. So this is what we call subarachnoid septum. So it is providing what we can say a posterior or dorsal support, posterior stability or support. Stability. It is something like it is ensuring that the posterior, it should not move beyond this. So in subarachnoid space, it should not, the subarachnoid space is like a sky, and your kite is, which is spinal cord, cannot go everywhere. It should remain within its limit. So that limit is defined posteriorly by this, caudally by your phylum terminal, cranially by your medulla oblongata, and posteriorly by your septum pellucidum. Now, sorry, not septum, subarachnoid septum, sorry, subarachnoid septum. Now let us see what is anteriorly is required. So in anteriorly also we have here, so if you see in this picture, so posteriorly here we got our subarachnoid septum, which we have seen in the previous slide. Now this one is your anterior end of it. So anteriorly you see there is a thickening you are seeing there. So that thickening, what you are noticing is what we call linea splendens. So what does literally linea splendens means? So linea means linea alba, linea aspera. Linea means your linea means thread. Linea means actually thread like. And splendens, the word splendens come from the word splendor. In English we have the word splendor. Splendor means something which is shining or glistening or shining. So that means this is a shining thread like structure. Literal meaning is shining or glistening, shining thread, which is what the literal meaning of your linea splendens means. So it is, what is a linea splendens? It's a pia mater gives off a septum in the anteromedian. So this gap what you're seeing, this gap is what we call as a anteromedian fissure. So this gap is called the anteromedian fissure. So the location of linea splendens is anteromedian fissure, which is the anterior aspect of your spinal cord. And in this area, a process is given off which become thickened. So you can see that white dot is a thickening of your pia mater in the anteromedian fissure, is what we call linea splendens. So what it does is actually, here you have a spinal artery. You will be knowing in a while. So in this area you have got a artery. So that is what we call as a anterior spinal artery. And around this you will be having a covering. So that covering is what we can call a thread like shining covering, which is extending from here, is what we call as a linea splendens, right? So in one way you can say that it is providing the anterior support. So anterior support to the spinal cord or stability to the spinal cord is provided by your linea splendens. So posteriorly by your subarachnoid septum, anteriorly by your linea splendens, caudally by your filum terminale. Now in this picture also we can see in this picture, so here we can notice this is spinous process, so this is posterior, so this is anterior. So in this case if you see, and if I mark it, so this is the area, this is your anterior and this is your posterior. So anteriorly what you have, these, you see there is a round area is there. Here you can see there is a round space. So you have this part, you can call this as a linea splendens. And here at the back you are seeing this one, this is what we call as a subarachnoid septum. So anterior posterior support is taken care, caudally is taken care. So if you go in this list, now we have the last part is there that we call as a ligamentum denticulatum. Ligamentum because there is a ligament. Denticulata is a plural for denticulatum, which means tooth-like. So these are the ligaments which are like a tooth. So how do you say tooth-like? So this is something like this. These are like your tooth. Like these are your tooth. So thereby ligamentum. This question came in the exam. So in this picture, if you see, sidewise, if you notice, you see these are the band which are coming in this side and this side. So these band, here you can see that's your ligamentum denticulatum. This one here, if I write it, ligamentum denticulatum are the lateral support or side support, and if you see they are present between, notice they are present between. Anteriorly you got your, this one is your anterior is this side, so anteriorly you have this one is your anterior root or ventral root, this one is your dorsal root. So it is lying in between them. So it is lying between them, and it is, the extent is, it starts from your pia mater inside, goes all the way between the two nerve root and goes, the yellow one is your arachnoid mater, it pierces the arachnoid mater, then goes into your subdural space, then goes to your dura mater. So it goes and attaches to the inner surface of your dura. So here also you can see in this case, so that's your ligamentum denticulatum here, and extent wise if you see, the extent is from your pia mater and goes all the way the dotted line is your arachnoid matter. It is piercing the arachnoid. Then green line is your dura matter. It goes and attaches to your, so thereby you can say it is triangular in shape. Apex is attached to the inner surface of your dura mater, whereas the base is attached to the pia mater. Yeah, so that is what is your extent and the attachment. Now location wise, it is present on the side or the lateral aspect of your spinal cord. Between here, you can see here, this is what we call anterior or ventral ramus. The back one is called posterior dorsal ramus. So it is lying between the ventral and dorsal roots. So it lies between, ramus is present here, but these are called roots. So this is your ventral root and this is your dorsal root. Both root combine together and form a spinal trunk which divide into ventral and dorsal ramus. So it is located between ventral and dorsal root. Now here also you can see, here if I draw it with this, these are your, you can see here, this is what is the base, this is the base and this is the apex, and that's your dura. In this case also I showed you here this part in the previous one, and this is a simple picture to represent a tooth-like extensions of your ligamentum denticulatum. And from this each extension, so this tooth-like extension what you are seeing laterally between the posterior and anterior nerve root from the pia mater, so these are from each band, so they are like one band. Each band, if you notice, each band will give rise to around 21 tooth-like processes which are, so these are all together, they are 21 in number. So 21 in number, they pierce the dura mater, and among this, the lowest one, which is, you see this is here on this side, you can see it is the lowest one is on this side, is of different shape. It is like a fork. It is like a fork. It is a fork shape, and that is how you identify that this is the lowest one. It is located between your T12 and L2. So location of this guy is between your T12 and L1 vertebra, and it is fork shape compared to other one which are tooth shape. This is fork shape, and in front of it you see you have the posterior root of your first lumbar nerve. So that means the L1 lumbar nerve located in front of it. So this is what is now this is the posterior view, so located behind it. So posterior root of first lumbar nerve located behind it. So what is the significance of this ligamentum denticulatum? So neurosurgeon use this place to do something called as a rhizotomy. So rhizotomy, rhizos means root, rhizotomy means cutting of the nerve root. So if they want to cut the sensory root, we call sensory rhizotomy. If they want to cut the motor root, they call as a motor rhizotomy. So in this case, if I want to do, if I take a blade, and if this is where I am cutting it here, so this is a posterior, so if I cut this one, this is your sensory rhizotomy, and if I cut this is a motor rhizotomy. Now how do I identify which is sensory and motor? So if this is your ligamentum denticulatum, in this picture is best, we can see here, and here you can see, and you cut open the spinal cord from the back, that means your vertebral column from the back, and you expose the spinal cord. And if you're placing your blade or scalpel in here, this is behind, this is here we are lying on, so it is placing behind this, so then you are doing a sensory rhizotomy. If you are placing in front of it, you are doing a motor rhizotomy. So when you do a sensory rhizotomy, the sensation carried by that particular nerve is lost. So sensory loss will be, so this is done when the patient cannot bear the pain at all, and where even your epidural anesthesia is of no use. So finally you have to go and cut the nerve root of that particular, so usually seen in the cancer patient. And motor is again where you don't want, the spasm is very high and the person is not able to carry out day-to-day activity, like your, I told you, scissoring gait in adductor spasm I mentioned in the lower limb. So in that case you have to cut the motor root. So you cut the motor root, and whatever the nerve impulse passing through it and going via that particular nerve, there will not be any action there. So this is what we do. So it is acting as a guide. So it acts as a guide for doing a rhizotomy. So wherein posterior to it we can do the sensory rhizotomy, and anterior to it we can do the motor rhizotomy. And how do they count it? So since when they open this at L1 and T12 and L1 vertebra, and where they will be able to identify the fork shape ligamentum denticulatum from here onward, because they know this is your 21st, now they can count, they can go up and they can count how they have to go. So that is how they can count and they, so it is something like your Lewis angle of your thorax, where they can use the Lewis angle to count the ribs. So it is something like a Lewis angle here, though the word is not used, but it is used, this fork shape last ligamentum denticulatum is used for counting the nerve root, so that a neurosurgeon can use this as a guide to reach a desired level to do a required rhizotomy. So this is about ligamentum denticulatum, very important, 3 marker and 5 marker. You write the applied part, everything. This is, so these are the 2 pictures which I have taken from Vishram Singh, the pink one, and this one also, the transverse section. This also you can draw it to show in the exam. So that finishes your meninges. So we are done with the introduction, we are done with the meninges, and now we go to the external features of the spinal cord. So the spinal cord actually begins now at this point. So what are the external features of spinal cord? You see the spinal cord has got, if you take the snake and if you observe from the front and the back, so you will see that it is not a simple snake, but there are so many lines are there on these. Now these lines are called as, if they are very deep lines, so they are very deep are there. So deep lines you call them as a fissures. That means they are very deep. That means you can actually see that something is can be kept. It is something like a gap between your 2 folds or 2 lines. Something like you can say between, what do you call, between the 2 buttocks, that space is there, that you call as a fissure. Yeah, that is a fissure. So between the 2 buttocks, that gap is what can be called as a fissure. So if it is deep, then you call fissure. But if it is superficial, if they are superficial lines and not so deep, only just the mark is made, so then these are superficial lines without any depth in it, so then you call them as a sulci. So you got, we know there are sulci and gyri in case of a brain, but in case of your spinal cord you have the sulci but you don't have a gyri, instead you got a fissure. So these are your fissures and sulci of your spinal cord, which are very important. Now these are important to decide or determine the anterior or the posterior aspect of your spinal cord. So in your neuroanatomy viva, if the spinal cord is given to you, they may ask you how will you identify which is anterior, which is posterior. So for that we will be will be using this fissure and sulci. And remember, if there is a fissure or there is a deep lines, a deep gap is there, so they are deep so that they can contain something. So this will have some content in it which can form as a identification feature for us to identify the anterior aspect. Okay, let us see. So we'll see this fissure and sulci here, how many are there and what are all there. And the attachment of the spinal nerve also becomes your external feature of the spinal cord. We also see that there are enlargements are there. You can see here the enlargement is shown, one in the neck region called cervical enlargement, other is shown in the lumbosacral region called lumbosacral enlargement. We'll see these enlargement, and also cauda equina I already mentioned in the initial part of it, and we'll go into the details of this. So let us begin. Now first we talk about the fissures and sulci. So don't worry about the crowdiness of this. This is very simple and easy. So anteromedian fissure. So there is one fissure and many sulci are there. Fissure is only one. So anteromedian. Antero means it should be present in the front and in the midline. This is the same area where we got our linea splendens, if you remember. So right in the linea splendens. So there the linea splendens, so there we have a deep groove. So you can say linea splendens is present in this deep groove, and this groove is what we call as, so I can show you the same picture here, and if I use the same thing here, and here you can notice, so this gap what you are seeing which is going in the deep area where I am putting the arrow now, so this gap is what we call as here antero, because it is in the front, antero, and in the midline. So anteromedian fissure. So anteromedian fissure is present in the anterior aspect of your spinal cord. And what is the extent? It extends all the way from your foramen magnum till your conus medullaris. And in fact, it goes beyond your foramen magnum and become your anteromedian fissure of your spinal cord also. Is it okay? So this one what you are seeing is, here we are, this is the posterior view. This is the posterior view. That is the reason we are not able to make out here. So okay, so an anteromedian fissure, so that's your anteromedian fissure. Here you can see, which is shown here. I even have one more here, you can see. So this is showing again your anteromedian fissure, the deep groove what you have in the front. And extent is all the way foramen magnum, and beyond foramen magnum also it is anteromedian fissure, but you call that as the anteromedian fissure of medulla oblongata. And here we call anteromedian fissure. So it continues above with the anteromedian fissure of medulla oblongata, and below Okay, so here I think the space is going. So